Imagine a world where biology and technology aren't just side by side, but they're actually merging, blurring the lines between what's natural and, well, what's engineered. Today, we're taking a deep dive into that very idea, our topic, biodigital convergence. And our mission, like always, is to unpack the sources you've shared with us, cut through the complexity, and really give you the most important insights, maybe some surprising facts too, about this truly transformative field.
Yeah, and at its heart, biodigital convergence is really where four massive fields are kind of synergizing.
Yeah.
You've got nanotechnology working at that tiny atomic and molecular scale, then biotechnology manipulating living systems, information technology, which is crucial for handling all the data and computation, and cognitive science, understanding the mind, intelligence, that sort of thing. This blend, we often call it NBIC, and that word convergence, it really means more than just mixing them together. It's about a creative union, not just of the sciences and technologies, but engineering, even people. It forces integration across fields that, well, used to be pretty separate.
What really struck me looking through the sources was how, well, established this idea actually is already. It's not some brand new concept that just popped up. We're talking about a framework that's, about 20 years old? Mihail Rocco and William Bainbridge first sort of laid it out back in 2003. And just think about how much information technology alone has changed since then. It's like Moore's law in overdrive, right? Doubling power constantly. Makes you wonder how the biological side has kept pace or accelerated to meet that. Okay, so how exactly does this powerful convergence, this mix of biology and tech, actually, show up? When we dive into the sources, it seems to boil down to two main pathways, both pretty fascinating. First, how digital tools are helping us understand life's mysteries, really decipher them. And second, how they're empowering us to, build or engineer new biological systems.
Absolutely. That first path, what we can call reverse engineering in this context, digital tech is just putting the pedal to the metal, supercharging our deep understanding of biological systems, and not just individual systems, but even entire systems of systems. It's pretty profound.
Yeah, what really jumped out at me there was this idea, you mentioned reverse engineering life's incredible journey, like 3.4 billion years. We're talking the oldest fossil cells from way back then, the multicellular organisms popping up around 1 billion years ago, oldest animals maybe 570 million years ago. How does being able to reverse engineer that kind of history change how we fundamentally see life? Are we seeing patterns or algorithms in biology we just couldn't spot before?
That's a fantastic question. And yes, that's precisely what these tools allow. To really understand these systems, we dig into what are called the omics fields. Think of omics as like a toolkit of powerful lenses peering into life. Genomics is the blueprint, the DNA.
Yeah.
Transcriptomics looks at how genes are actually used, expressed. Proteomics maps out all the proteins doing the work. Metabolomics shows us the small molecules involved in everything. And then there's epigenetics, how gene activity changes without changing the DNA code itself. And microbiomics, studying those huge communities of microbes living with us and, everywhere. All these descriptive fields then feed into understanding the bigger picture physiology, the whole life cycle through things like developmental biology, evolution. And we're even broadening that out now, looking at environmental interactions, larger systems. So exosomics, what we're exposed to, understanding whole ecosystems, how they evolve, maybe even that bigger Gaia concept.
That amount of data for just one person it's just mind-boggling. The sources mention the human genome, right? About 3 billion base pairs. That's like 700 megabytes of incredibly complex code, plus mitochondrial DNA, the proteome with maybe 20,000 proteins, the metabolome, over 110,000 molecules, and the microbiome, an absolutely staggering 316 million genes.
Yeah.
So how does managing that sheer scale actually impact researchers day to day? Even with sequencing costs dropping so dramatically, making it possible to get all this data, are there new bottlenecks?
That's exactly the point. Yeah, sequencing costs have plummeted. Fantastic. We can generate the data. But the real challenge, the bottleneck, has shifted. It's now about the analysis, the interpretation. We're drowning in data in a way. We need totally new computational tools, AI, new ways of thinking to find the meaningful patterns in what's basically a universe of biological information. It's not just about getting the raw data anymore. It's about figuring out what it means for medicine, for agriculture, for materials, you name it.
It's incredible, the depth of understanding we can achieve now. But, okay, what about the other side, the creating side? Re-engineering things for new purposes. Where are we seeing this forward engineering part of biodigital convergence really take off?
Oh, it's really accelerating. This is where digital tech basically turbocharges the engineering side of biology. It enables incredibly powerful new bioengineering capabilities. We're talking about, well, everything from designing artificial organs that could save lives to creating sophisticated little biobots that might deliver medicine right where it's needed. Think about smart farming, using AI and sensors to boost crop yields dramatically, or the potential for human augmentation, things like brain-machine interfaces. Could they restore lost function or maybe even enhance cognition? The scope is just huge. It stretches from the core stuff like biochem and bioinformatics right into really cutting edge areas. You mentioned BMIs, cyborgs, exoskeletons, biohacking, organ on a chip tech, GMOs, even using sensors for tracking epidemiology.
Right.
It's vast.
That's an absolutely astonishing list of applications. But, you know, as you describe this landscape, this rapid convergence, it immediately brings up a critical question for me. How do we make sure all these incredible innovations don't just stay in their own little silos, or worse, create unforeseen risks down the line? And I guess this is where the often uh, maybe less glamorous, but totally essential work of standardization comes in.
Precisely. Couldn't have said it better. That's exactly why groups like the IEC Standardization Evaluation Group, specifically SEG-12, exist. They focus entirely on biodigital convergence. Their main job is to look at these super complex systems, figure out everyone who's affected, researchers, industry, governments, the public, find the standards we already have, spot the crucial gaps, and then suggest ways forward. Architectures, roadmaps for future standards. And it's important to note, SEG-12 is what they call a time-boxed project. That means it has a deadline, usually 18 to 24 months. The goal is to quickly map out these unexplored areas and recommend how the IEC should tackle the standardization work needed.
And their outputs sound critical. First, you mentioned they're working on a common language, a shared vocabulary, which, yeah, you can just imagine how vital that is when you have neuroscientists trying to talk to data scientists, trying to talk to materials engineers. And second, they produce this green paper, which is like a public preview, a technology report that anyone can access.
Exactly. Freely available. It dives into the tech aspects and touches on those really important ethical and social issues, too. It's meant to get the conversation going, really.
Okay, so to give everyone listening a real taste of how deep this goes, let's maybe explore some of the specific working groups within Sec12. What frontiers are they tackling?
Sure. Let's start with the frontier of reverse engineering living systems. That's mainly Working Group 2 or WG2. Within that, when we talk omics, again, genomics, proteomics, all that we're dealing with, as we said, just massive amounts of data, big data. And there are already efforts underway to standardize things like massively parallel sequencing, how genomic data is processed. That's vital just to manage it all. Then you have synthetic biology. This is really about creating new biological parts, devices, or even redesigning whole systems for useful stuff. Applications are everywhere: biodetection, making biofuels, biomanufacturing. People are even looking at DNA for data storage. But here's something interesting from the sources. Despite all that potential, standardization in Symbio is still really early days. They describe it as an incomplete system, less coverage, no counterpart TC, which basically means the standards are seriously lagging behind the tech breakthroughs. That definitely creates challenges for actually using this stuff reliably and widely. And the third big area in WG2 is neuroscience. Studying the nervous system, brain, spinal cord, nerves, the goals are huge. Understand how the brain works, find cures for awful neurological diseases, maybe even simulate or build brain-like machines, AI, brain organoids. So the question that pops up immediately is, what are the standardization gaps? And they're pretty significant. Things like input-output systems, data formats, protocols for experiments, how data is processed, definitely ethical considerations, clinical guidelines. The sources give a specific example, needing standards for just preparing samples for 3D optical imaging of neural systems. Without that consistency, how can you compare results from different labs? It really slows things down.
And it's worth really hammering home who needs these standards, right? Because it's not just scientists and labs, it's the companies building products based on this. It's academic researchers trying to build on each other's work. It's governments making sure things are safe, business developers creating new markets. And maybe most importantly, it's you, the listener, the users, the individuals who stand to benefit, but also need to understand and, frankly, have protections around these powerful technologies.
Well said. Okay, let's shift gears to Working Group 3, WG3. This one looks at standardization opportunities directly in life systems and bioengineering. Take biosensors, for instance. One cool concept is the bioelectronic nose. The idea is pretty neat. Use an olfactory receptor, that's the biological part that detects a smell or chemical, and combine it with a transducer, something that generates an electrical signal when the receptor detects something.
Wow, okay, a bioelectronic nose. The applications, you can immediately see them, can't you? health diagnostics, maybe in wearables, detecting illness early, food quality telling you if something's gone bad, public safety, detecting viruses, toxins, explosives, environmental monitoring, air, water quality. That's huge. So the standardization targets there would be what? The receptor itself, the transducer, how the signal is processed, the pattern analysis, making sure all those bits work together reliably.
That's exactly right. Another really fascinating area WG3 is digging into is human digital twins. Imagine having a dynamic software model of you, a virtual copy that collects your real-time biological data, simulates your body's processes, essentially a digital you. The potential is mind-blowing, predicting health issues, personalizing treatments down to the individual, maybe even testing new drugs safely in your digital twin before they ever touch your actual body. Now, connecting this back. Getting harmonized, interoperable data standards for these digital human twins is absolutely critical. Non-negotiable, really. The source material had a great analogy. Think about fire hydrants. Historically, there were like 600 different types of hose couplings across different places. Imagine the chaos in an emergency. Disparate data formats for digital twins would be just like that, hindering progress. So standards are desperately needed for data formatting, the predictive models, how we exchange phenotype data, tracking data provenance. WG3 also covers artificial organs and organoids. These can be mechanical, biomechanical, or purely biological constructs. Standardization needs there are massive. Everything from the source of the cells and how they're prepared, to quality control, function tests, safety, risk assessment, even the materials used, like the specific bioink for 3D printing organs, plus all the mechanical and electrical systems involved if it's not purely biological. Then you have things like CAR cells. That's a type of genetic engineering, mostly used for cancer right now, but expanding to other diseases. Standardization there currently leans on existing guidelines. Good laboratory practice, good manufacturing practice, good clinical practice, plus national rules. But the gaps identified are in areas before commercialization, like preclinical work, safety of the viral vectors often used, handling in the lab, and data standards again. And of course, we have to mention CRISPR technology. The genetic editing tool with, well, almost limitless applications across all sorts of biotech, green for agriculture, white for industry, red for medicine, blue for marine. It does have some existing standards, like vocabulary and standards for synthesizing nucleic acids. But potential areas for more standardization include common criteria for judging product performance, identifying things that affect performance, guidelines for sample prep. Consistency is key. And underpinning all of this absolutely fundamental is data quality. All this incredible data we're generating, it's only useful if it's high quality. Think of it like cooking. You need good ingredients. For biodigital data, that means it has to be available, usable, reliable, relevant, and presented clearly. Good presentation quality. This isn't just a nice to have, it's essential for any application. And it's critical for adhering to what people call the FAIR principles, making data findable, accessible, interoperable, and reusable. Without a good data quality, obeying FAIR principles, even the most amazing science can just, well, stall out. Okay, moving on to Working Group 5, WG5. This one zeroes in on agricultural systems, and that's a huge area of forestry, fish farming, aquaculture, livestock, the new cellular agriculture, even molecular farming where you use plants or animals to make pharmaceuticals. There's a direct link here to that big UN Sustainable Development Goal, zero hunger. This includes Internet of Things applications like precision agriculture using sensors and data, and also the genetic engineering side of food production.
Wow, okay, but the sources mentioned WG5 is facing some real headwinds. Small number of people involved, low attendance, competition from other groups like ISO's smart farming work. That sounds like trying to organize a crucial project with, like, half the team missing and another group doing something similar next door. What does that mean for the future of our food systems if this standardization work struggles?
That's a really good analogy, and yeah, the implications could be pretty significant. Which brings us back to who needs these standards in agriculture? Well, pretty much everyone involved. It's crucial for genetic engineering, for plant and animal breeding, for digitizing all the farming processes, for using remote sensing data effectively, for integrating all that diverse data to actually do precision agriculture, and for making supply chains more efficient, tracking things from the seed all the way to food, animal feed, or fiber. But as you hinted, one of the biggest hurdles is data itself. There seems to be a general reluctance maybe to standardize how data is acquired and shared. Makes it hard to integrate. Plus, you have basic infrastructure problems like patchy rural internet, and then the really thorny issues around data ownership and monetization. Who actually owns the data coming off a sensor in your field? Who gets to profit when it's analyzed and combined with other data? These are big unresolved questions.
And the supply chain challenge, too. You mentioned Walmart tracking melons with blockchain years ago, which sounded promising, but the sources say wider standardization efforts there are still pretty rare. Is it just too complicated, the supply chains themselves, or is it like companies wanting to keep their data and methods secret for a competitive edge?
I think it's definitely a mix of both. Global supply chains are incredibly complex, no doubt about it. But yes, competitive advantage is a real factor. Companies often see their supply chain data and optimization methods as proprietary. Looking ahead, the future here really needs improvements in managing these massive genomic data sets from breeding programs and the sensor data from farms. It needs better ways to integrate diverse teams, data scientists, statisticians, biologists who often speak different languages. It needs that interoperability between different systems. And yes, it absolutely needs progress on resolving those data ownership questions. It's a huge job.
And finally, it's really important to mention there's a dedicated group, WG7, looking specifically at the human side. The crucial social aspects, ethical concerns, risk, resilience, safety management, all related to biodigital convergence, including data governance. It's definitely reassuring to see that the human element, the societal impact, is being considered right from the start in these standardization efforts, not just bolted on later. So there you have it. You've just taken a pretty deep dive with us into this incredible world of biodigital convergence, a field that isn't just some far-off future thing, it's actively shaping our present, right now. From how we understand the absolute basic building blocks of life to how we're engineering totally new biological systems for health, for food, for environmental solutions.
Yeah, we've really seen how digital tech is boosting both sides of the coin. Reverse engineering life to understand it, and forward engineering solutions based on that understanding. And we've seen why global standards, like the work IEC Seg 12 is doing, are just so critical for safety, for making sure different things can work together, for ethical progress across all these diverse applications, from bioelectronic noses to human digital twins to making agriculture more sustainable.
It's definitely clear this convergence is moving incredibly fast, maybe faster than we even realize sometimes.
Absolutely. And that speed, that power, brings us to a final thought, maybe a question for you to chew on. As our ability to blend the biological and the digital keeps growing, keeps accelerating, how might the very definition of life itself start to change or continue to evolve?